Hello.
One second, I'm calling you.
No, only la face na anga tali போய் பேசுங்க.
Okay, okay. Start பண்ணிடலாம்.
Okay, 10 minutes ஸ்டார்ட் பண்ணிடுவோம். பண்ணதுக்கு அப்புறம் சொல்றேன். Hello. Can you hear me?
Yes, sir, we can hear you,
Yes,
but
sir.
your video is off.
Oh, okay.
It's
Actually,
okay. You want
you
to
switched
just
off.
check the slides, sir?
Yeah. One time, if I can check my
Okay.
slide, it'll be good.
I will stop share. Already we are in live. I will stop my share so you can start sharing your screen.
Okay.
Okay. Once you stop,
Or
again
I
I'll
think,
rest.
or if you're already in live, I think you can start telling me. So you muted my video, right? So I don't need to show my video.
No, sir. While on live, we will. I will ask you to resume your video.
Oh, okay. So I can wait, right? Whenever you introduce me, I can start.
Good morning, one and all. Before we start this webinar, we would like to give you some basic instructions to be followed during this webinar series. Kindly do not chat as soon as you log in, giving greetings and introducing yourself. Your name will be correlated with your mail ID in registration form and will be recognized accordingly. Do not ask questions as soon as the sessions begin. You can ask questions half an hour after the start of everyday session around 11 a.m. Your participation in all the 3 webinar sessions is important for us to be eligible for certification. So kindly log in before 10:45 a.m. as the chat room will be locked after the set time. End of the session, feedback form link will be posted in the live chat on YouTube. So it is mandatory to fill the form for every session to be eligible for the e-certificate. We seek your kind cooperation for the success of this webinar series, which would be starting in a while from now. So kindly stay tuned. Thank you so much.
A very good morning to one and all. Greetings from the PG and Research Department of Biotechnology, Women's Christian College. I welcome you all on behalf of my department for this webinar series on global research trends on COVID-19 opportunities and challenges. I also want to welcome our guest speaker for the day, Dr. Namashivayam Ganesh Pandian from Kyoto University. Thank you, sir, for accepting our request to be the guest speaker for the day. Everything is possible only with the Almighty's blessings. Dr. Julia Harriet Sumati would lead us in prayer, followed by Dr. Anita Ajay Singh, who will introduce the guest. The question and answer session would be handled by Dr. Anchana Devi. I hope we all have a very fruitful learning experience. Jai Hind.
Let us pray. Loving Lord Jesus, we thank you for this beautiful day, O Lord. Lord, as it says in Psalm 118, verse 23 and 24, this is the day that the Lord has made. We will rejoice and be glad in it. Yes, Lord, we thank you for giving us the grace to see this day where our webinar series begin. Lord, first and foremost, I would like to thank our dear principal, Dr. Lillian I. Jasper, and the management for all the help, Lord, they have rendered to us by way of their support for the series. Lord, I pray and thank you for the student community of Biotech Department, spearheaded by Ms. Yashashwani and Ms. Shamala for their tireless work and endless effort they have put in to make this event happen. Lord, I pray for all the participants who have registered here. The students have registered with an urge to learn new things, O Master. Bless them. May the session be a fruitful one. I pray especially for our guest speaker for today. Thank you, Lord, for bringing him in our midst and for connecting with us. Lord, I pray for his research career. Lord, bless the work of his hands. May he prosper. May he come out with new findings to combat this dreadful disease. We commit everything into your nail-pierced hands, Lord. Lord, we pray that your spirit will hover from the beginning till the end and that it will guard all so that there will be no disturbance and we have a smooth lecture. We give you all glory and honor. In Jesus' matchless name I pray. Amen.
Good morning, all. I deem it a privilege to introduce Dr. Namashivayam Ganesh Pandian, who is a principal investigator and junior associate professor at the Kyoto University, Japan. His other affiliations are with the AO Research Institute, Switzerland, and NCBS InSTEM India, where he is an honorary visiting scientist, and Rutgers University, USA, where he is a visiting professor. Dr. Pandian holds a doctoral degree in the field of biosciences from Niigata University. His master's and undergraduate degrees are in the biotechnology and biochemistry fields, respectively, from the University of Madras. His research interests are in nanobiotechnology, epigenetics, cellular reprogramming, bio-inspired therapeutics, and artificial genetic stitches. His research areas include nanomaterials, cell biology, genomics, therapeutic gene modulation, nature-inspired biomaterials, biomedical engineering, and the list goes on. Dr. Ganesh Pandian has received several awards. I will mention the recent few. TEDx Kyoto University Speaker Award in 2018, 8th World Congress on Targeting Mitochondrial Research Excellence Keynote Speaker Award in 2017, Brilliant Publishing Organization's Rising Star Award in Science in 2017, and many more. Dr. Pandian has, to his credit, over 85 publications in reputed journals. He has published 3 books and has made over 21 presentations all over the world. He has 8 research projects to his credit. During this COVID-19 outbreak, Dr. Ganesh Pandian is in the limelight for his indigenous breakthrough in the detection of the dreaded virus. Dr. Pandian, along with his associates, has invented an X-ray based software which can rapidly screen the virus with a 99.69% accuracy. It has a rapid screening capacity of 100 images in 3.63 seconds. This software can detect and differentiate positive and negative attacks. We cannot imagine a better scientist to address us on this vacillating virus. So now over to Dr. Namashivayam Ganesh Pandian.
So hello everyone. So probably my video is muted, I suppose. So probably you can't see me, but I think you can see my slides. And so first of all, thank you one and all for having me here. And first of all, I'll be very thankful to Dr. Anjana Devi for reaching out to me to talk about the COVID-19, the virus with the vacillating genetics. So this is the topic given to me. This was not the topic I chose to give, but this is very important, I suppose, because there have been a lot of hoaxes going around the world about this virus getting mutated and maybe whether the vaccine will work or not. Probably this particular topic will be more interesting now, and probably I can clarify some of the possible situations that is the current trend in all over the world. So first, I would like to start by talking about the place I live in. So I live in Japan. So I live in the ancient capital of Japan, Kyoto. So Kyoto is the Japanese are known to face a lot of natural disasters. Of course, COVID-19 is kind of a natural disaster as of now. And so this kind of natural disaster, Japanese have been, getting exposed to this kind of natural disasters now and then. And they always rise from the ashes, like the phoenix. So the bird on top of the this beautiful golden temple, which you can see. So this is kind of the landmark of Japan. Like in India, if you come, people take a picture in front of Taj Mahal. Likewise, in Japan, usually people prefer to come and take a picture in front of Kyoto. It's a very beautiful place, and so people rise from ashes So that's what the symbolizing the Japanese spirit. And what you can see here is the beautiful forest, bamboo forest. So this is listed as one of the CNN's top 100 places one should visit before they pass on. So this is a very beautiful place, and please do remember, whenever you come to Japan, try to visit Kyoto. And this is where I work. So this is our institute name is Integrated Cell and Material Sciences. It's a part of Kyoto University. And previously it was part of the World Premier International Research Center Initiative, which is one of the proposal given to the mixed Japanese ministry. So what we do is we try to break the barriers in sciences. We try to, before 100 years, we were not known as the, like biologists, physicists, biophysicists, and so on, right? So those days, people were known as scholars. And even the field of genetics was actually invented by a mathematician and a priest, Dr. Mendel. Now this particular program aims at, breaking all the boundaries, and we try to fuse the sciences. So there is no barriers like chemistry, cell biology, or physics. We try to fuse all these barriers, and we try to make a fusion science. So we work on stem cells and mesoscopic domain, which I will talk about in later, because people now are more interested in not in curing diseases, not in regenerative medicine. People are more interested in coronavirus. But I'm very happy and proud to say that our institute got affiliated once with Professor Dr. Shinya Yamanaka, who got a Nobel Prize for his discovery in induced pluripotent stem cell in 2012, and recently Professor Tasuku Honjo. So he is the current deputy director of our institute, Kyoto University Institute for Advanced Study. So he got the Nobel Prize for immunotherapy. And I've been blessed with good team members. So whatever I am is because of the team members which I had and is having. And I just want to show this slide of some of my alumni members and the current members. So we are basically gene switchers, and you can notice that most of the places, even in my affiliated lab in Switzerland, there are a lot of women there. So I think, and I have 2 daughters, so probably that qualifies me as a speaker here in Women's Christian College. And I believe strongly in women power. Okay, so what I do, you may wonder, who am I, right? So I'll just say a very simple introduction about what the work I'm doing. So I am trying to decode the language of bacteria. So, we talk, now I'm talking in English, most of you talk in your local language. So what is the language of the bacteria? Bacteria do not have any organs, right? How do they communicate? They communicate by releasing the chemicals. So I'm working on a chemical called pyrrole imidazole polyamide, which is basically a heterocyclic compound derived from the bacteria. So these 2 compounds called P and I, shown in blue circle and red circle, can actually talk with the 4 letters DNA, 4 letters of the DNA like A, T, G, C. And using this molecule, what we do is we try to switch off the bad genes and switch on the good genes. That's what we do. So more than that, I can talk if we have time after this. But what we try to do with this technology is that we try to, give kind of a GPS to the epigenetic drugs or anti-cancer drugs. We try to make an anti-cancer drugs out of this technology. We can also visualize the telomeres and other kind of, important sequences inside the living cell. And very importantly, which the work I'm proud of, is that we are now currently developing this technology as a kind of a mitochondrial drug. And we're also using this to regenerate the lost body parts. We try to do it in the petri dish level at this moment, try to convert one cell to another. Okay, so basically this is what we do. We try to switch on the good genes and switch off the bad genes. That's all you need to know at this moment. I think more than this, whatever I say, you are not interested these days, right? So all you are interested in these days are on viruses. The whole world is now interested in viruses. So what are viruses? viruses, the term is always associated with the something bad. But you have to know that the human genome is full of viruses. So we evolved with the microorganisms and viruses in our body. So how many of you know that mitochondria, as you know, is a powerhouse of the cell, was once a bacteria. During evolution, it got internalized into the cell and became the powerhouse of the cell. Likewise, viruses also got inside our body and have been evolved over the our evolution, and a lot of other kind of microorganisms are also getting inside our body. And what we are is because of this recombination and co-evolution of the microorganisms with us. Probably you also know that our body itself, 10% of our genome is made up of the microorganisms. So we also have microorganisms inside our body. So viruses are always not bad, but usually why it is considered bad, probably I will talk about that later. But one thing you may be surprised about viruses is that, one of the major feeling people experience is love, right? So how many of you know that love is proclaimed to be a virus? So this is a beautiful article written by Carl Zimmer. Later you can go and Google more about that. I don't want to, say more detail, but what I can say here is they showed an experiment in the male prior balls, the kind of a mouse you can say, and they showed that the expression of one particular vasopressin V1A receptor was able to make the the brains of the male prior voles to have more love feeling than the bearded voles. So it's a very beautiful article. So the whole concept here is that love could also be because of the virus. Okay, so that is one of the theory that scientists have been proposing. And I'm telling you here because these are all the cohabitants of our earth. And probably some of you know about the Game of Thrones, the series. So you have to know that the coronavirus is just the beginning. And there are a lot of ancient, never before seen viruses that has been, getting, the man is actually coming into contact with the new viruses from now. So this particular viruses could be because of the natural selection. That's what is believed now. But there is a possibility that some of the unknown viruses can come because of the global warming, like the Tibetan glacier, getting melted. So we may come into contact with several new viruses from now on. So the winter is coming. So are you prepared? So that is the question now. So, but don't worry about that. The most important thing at this moment is not to get panicked, to know how to have a sustainable coexistence. So before talking about the viruses, I want to say about the journey, which could be interesting for you also to know why I became a scientist. So I became a scientist because of the one lockdown, which happened in my class 9. So I had a problem where I was not able to see the sunlight. And at that time I was locked down in my home, and I used to always ask my mother about I just want to engage her in some conversation. So I always ask why. And then when she tried to answer, I asked why not. So I like questions, because I just wanted to make a conversation engageable. So this is what I try to do. So one time, I usually get quickly infected whenever I go and play outside. So I played with a bug. I had a pet bug with me. And one time, I was playing in the garden, and I got sick, but bug did not get sick. So I felt very bad. So why, I'm not, I'm getting sick. Is my immune system that bad than a bug? So then it actually, raised me in a question about, do bugs get sick? Or why are they immune to bacteria? So that was my question then. Probably, this could be a question you may also think now. Because this kind of the bugs, like the cockroach and all, they live in a very, very bad environment. You know that, right? So they live in a very contaminating environment, where if we humans go and be there, we will get fever and even whatever the kind of stuff we have. So then I came to know during my bachelor's that, they do get sick. In fact, people have been using the biopesticide called Bacillus thuringiensis. That was my PhD project. So they can kill the healthy silkworm, they can kill the bugs basically. So insects do get sick. That I understood. And so this was one of the example of how the bacterial biopesticide, which is the Bacillus thuringiensis, can kill the insect. Then I had this question: do bacteria get sick? Can bacteria get sick? So that was the question. And then I learned about this beautiful technology, which has been probably most of you know, that is CRISPR-Cas9. So what is CRISPR-Cas9? Probably you already know about this, but I can show a very simple illustration for the people who don't know. Bacteria, they also live in a very bad environment, and they are more prone to the virus. So the virus, the whole purpose of the virus, so there has been a huge controversy whether virus is a living thing or a non-living thing. But the purpose of the virus is to multiply. That's all they need actually. So what they do is always they try to insert the genome inside the host cell, in this case the bacterial cell. But bacteria have a defense mechanism called CRISPR-Cas9, with the enzyme called Cas9 can actually cut the specific DNA, the specific sequence, nucleic acid sequence, which can be DNA or RNA. And in this case, CRISPR goes and recognize and then they cut the virus out of the system so that they basically have an immune system. So this CRISPR-Cas9 can be used to rewrite the misformed letters in our body. So we are all made from letters. I'll talk about that in detail later. But the people have been using this CRISPR-Cas9 for gene editing basically. And this particular SARS-CoV-2. So I tried not to, say things which you already know. I try to say things which you may not know or, could be interesting in my perspective. So I'm basically a genetic engineer. So I want to talk more about genetics here. So SARS-CoV-2, the virus, probably you have seen the picture many times enough, right? So basically they are made of 30,000 letters. So we always, look in my lab, we always look into any living organisms in terms of letters. Okay, so they are made from 30,000 letters. So this is the genome organization of the virus. Please remember about this RNA-dependent RNA polymerase. So this is nothing but the copy machine of the virus. So they make this copy machine. So they make this protein to copy their genome into the host cell. And so there are some facts which you could be interested in. So unlike the other countries where people, the older population are getting more vulnerable. So in India, relatively, the most infected cases are within 20 to 49 and the age of 20 to 49. So you have to be careful. So please try to be careful and don't, this is a new virus. People are learning. So whatever I say today can turn out to be, not so true tomorrow. So people are learning now. So please try to, adhere to your, please listen to the guidelines given by the WHO, CDC, and our ICMR. So one good thing at this moment, at the current trend, is that the virus is less hostile towards the female. So relatively, the number of infections are more towards the male. 76% of the male are infected, but relatively less female are getting infected. So you have to be happy about that in a way. But still, please do take the precaution and try to be careful if you have a pre-existing condition like diabetes, heart and kidney disease. Probably you heard about this from many other sources, so I don't want to repeat those situations again. I'm interested in history always. I always want to know how things started. And this is how the coronavirus is basically a virus, a large a family of virus. It's very distinct from the others. So people have been studying this virus, and this was first reported in 1980 in the first International COVID Symposium, in detail. So in early in 1900 itself, they know about this virus. That's how they gave the name coronavirus based on their the royal crown looking structure. And it got noticed by everyone in the early 2000. because of the SARS-CoV and later MERS-CoV, and now the SARS-CoV-2 is the cousin of these viruses. So all are known to be originated from the bat, basically. So people already know about SARS-CoV in 2002. This was actually more, deadly than SARS, and MERS was relatively more deadly than this SARS-CoV-2. And that time, the people know that the intermediate host was cuvette hat. The two genomes, they got recombined, and then they went on to the host humans. That's how it started. All this thing started. And at this moment for SARS-CoV-2, people have been hypothesizing that the pangolin, the anteater pangolin, in some way, this particular bat, this particular gene from bat CoV, rat GL3, G13, and then the pangolin, they got recombined, and then they passed onto the humans. So that's what is the current belief now. And so beta coronavirus has been, is known to be infecting many other, living organisms. Like, they are known to be, infect even the giraffe, lion, and deer, and a lot of animals. They are known to be, they can infect. But this particular beta coronavirus, the SARS-CoV-2, have more affinity in a way, more affinity than the previous SARS-CoV towards our human body. So again, this paper from Zero et al. In Nature, so they indicate that this could be because of the natural selection. So just because of a small difference in the amino acid, so the AC, the spike protein have relatively more affinity towards the ACE2 receptor. So that's how they, kind of infect the human. And as of now, about the situation. I don't want to go in detail. A lot of, it's kind of the pandemic already. So all over the world, people are getting infected. And one good thing you have to know that people are also, as of now, there are no vaccines. There are no approved medicines, apart from some of the emergency approved medicine like remdesivir. But still, people are coming out of the coronavirus. So that is one of the good thing we have to think about this. So it's not like when you get this virus, it's a death sentence. Not like that. So many people are coming out of that, and relatively the death rate is not so bad. So please don't panic. We just need to understand the virus. And I think scientists are working hard on that. And here I want to say one of the work which we have done in our lab in combination with the Indian Institute of Technology, Roorkee. So, this all happened because one of the postdoc, Dr. Zotao Yu, so he went to China. So he's a Chinese in origin, and he's my employee, and he went for his holiday to China. So, and he came back before, they shut down. But one thing he said is that they use the artificial intelligence, and they use this CT scan. So, you can see that in one day, for a lot of spike, about 15,000 patients were, tested in China here. And then after they tested, they efficiently quarantined, and then they kind of brought down. So again, so China did not invent any vaccine or any medicine, but still they came out. They almost, flattened the curve. So how was it possible? It was possible because of the 3 T's: testing, tracing, and treatment. Treatment is still under works. Tracing, as you all know, people with the modern technology, people have been, tracing using the smartphones to quickly determine where they are using the GPS and all. But testing. So testing, as you know, RT-PCR is the most known method, but it's quite expensive, and some spaces it's kind of constrained also. And the rapid antibody testing, the efficiency is not so high. So there was a huge demand of some technology. And then one of the brilliant student of my lab, Mr. Vinod Joseph. So he told me this idea about why don't we use chest X-ray images instead of CT scan? Because people, when they use the CT scan, the way the CT scan is taken is when the patients can go inside the CT scan, and that itself can be a reason of spreading of coronavirus, because it's very difficult to disinfect and all. So we wanted to try to as much as minimize the exposure of the patient to the healthcare workers. So instead of people using PCR as a validator and use the X-ray as the tracer, we wanted to use the X-ray as the kind of a surveillance tool, and then determine who can be tested for PCR. In this way, we can save a lot of money. So here I have to thank my collaborator, Dr. Professor Balasubramaniam Raman and his team in Indian Institute of Technology, Roorkee. They were the one who did all the work, and we were just telling them what can be done. And so this was the proposal which was made by me. And so what I said, maybe we can have a representative bus, as you can see here, and then we can have the X-ray machine where the social distancing is maintained. And then, we can change the, we can transfer the data and then do this kind of, machine learning where, people, where the machine, the software package is shown the images. Okay, this is the COVID positive, and this is the COVID negative, and this is the viral pneumonia positive, because, it's very easy to distinguish between coronavirus and the normal patient. Just, sometimes even we don't need artificial intelligence. But why this is more important to use artificial intelligence is because it can also, overcome the false positive, like viral pneumonia. So we use this as we propose this as a surveillance tool. It's very good for the post-lockdown situation, and it's far superior and medical driven than the thermal scanner. People are now, checking the temperature. And one thing which is, we notice is that which was also proven by the recent papers, not by us, but from the other communities around the world, is that even some of the asymptomatic patient, they can have some changes in their lungs, which can be determined at the early stage. And related to this, there's a very minimal radiation exposure here. So we were able to distinguish between the COVID infected, healthy, and pneumonia, and we were able to do it very fast. And probably some of you may have seen this news, like that was covered in Times of India, that appreciating our bilateral relation. And I have to thank our Consul General of India, Osaka Kobe, Dr. Sham, for supporting this friendly collaboration. And probably you also would have seen me in television, particularly in Tamil television. And so we offered the X-ray technology free of cost. And meanwhile, many people also, even in IIT Roorkee, there was another team who developed this kind of technology. So it doesn't matter at this moment about who developed the technology, but the government took this, the IIT Alumni Council took this. You can see the model of bus. And our chief minister also unveiled some of the mobile X-ray unit. So we are glad that our idea was taken, and ours and many other people's ideas were taken. And so that was my contribution to coronavirus at this moment. So yeah, test tracing, you all know, testing, this is one of my contribution, and not just my contribution, it's a team effort again. I have to thank again Dr. Balasubramaniam Raman of IIT Roorkee. And moving on to the treatment. So why, treatment of coronavirus is tricky? Why can't we find a drug? Are we not having a drug? We do have drug. It's not like, we don't have any drug. We do have drug. There are many drugs which are known to, stop the entry of the coronavirus. Either the entry, like, inhibit the viral entry, like, Camostat, mesylate, or Orbidol, or the famous, hydroxychloroquine. People are, known to, after President Trump, of his own admission that, he is consuming this hydroxychloroquine. It's been, widely criticized. We still don't know. It's still early. But as of now, the studies show that it's not so effective, but it's effective in some cases. It's been under controversy. But anyway, there are many drugs available now. So there are no drugs for coronavirus, but there are drugs which can, alleviate the symptoms of coronavirus. So the most important drug, that goes into the core is to stop the RNA-dependent RNA polymerase copying of the viral, the coronavirus. The mechanism of action is that they copy their genome by coding the RNA-dependent RNA polymerase, and then they try to make a copy of the RNA in the host cell. This is an RNA virus, and so this particular copying can actually, cause some mutation, which I will talk about later. So they try to inhibit this process by using the drug like remdesivir and favipiravir. probably you know about this tablet called Avigan. Avigan is a tablet developed by the Toyama Pharmaceuticals in Japan, and remdesivir is the emergency approved drug from FDA recently. And other virs and the chloroquines are all under work now. And so there are some drugs already which has been showing promise. But why people are not using this is because we have to be very careful. As I told, even with the drugs, people are coming out of coronavirus. So we have to be very careful. But whatever the drugs we are giving is not only killing the virus, but it's also very safe for consumption. Hydroxychloroquine are known to increase the long QT of heart. So we have to be very, very careful in administering these drugs. So please don't take any drugs without a medical prescription. And about the antiviral drug remdesivir, so this is the mechanism where the SARS-CoV, they kind of recognize the ACE2 receptor inside the host cell, and then they put their RNA, and that RNA gets copied and then into the human genome. So that is inhibited by the remdesivir, this drug. And so this this has been shown to be very promising, and that this already entered the phase 3, and people are now working on the randomized clinical trial. And I hope that this comes out well. But one thing we have to be very careful here is that the coronavirus, this particular virus is mutating. That's true. But whether that mutation, this particular coronavirus have this proofreading system, so they can actually be very careful in not making this to be materially different. So I personally believe that the remdesivir and this particular favipiravir, because of the ease of the structure, these also follow the very similar mechanism to stop the replication of the virus. So this favipiravir, you can see the structure is very simple. This can be made relatively easy at cost, and in fact, Japanese government stockpiled about 2 million of this drug for emergency use. But one one word of caution is that Avigan is known to cause some birth defect. So this has to be administered again only when people have severe symptom. So people are now working on whether this is going to be safe or not. But I am counting on my thing on EIDD 2801 from the Emory Institute, because that particular drug can bypass the proofreading system of the coronavirus. So about the immunity, so the immunity is now the most abused word, right? People have been telling we have to increase our immunity, we have to increase our immunity. So we have to be very cautious that we should not increase our immunity, we should control our immune response towards the virus. So that is the actual term. Otherwise, the actual people who are dying is because of the cytokine storm. Our immune response get hyperactivated at some point after becoming stressed that because of the awareness that corona is infected. So naturally, because of the infection, it can cause the immunity, and by the artificially by the vaccine, we can have the immunity. And even the mother's milk, in this case also the maternal antibodies can cause the immunity. And recently, about this Israel, they invented this monoclonal antibody that is also very good for the immunity. But we have to be very careful that neutralizing antibody, although it's been showing very promising success, whether it can be scalable, whether it can be made available for 7 billion people on the earth. That is the big question at this moment. And of course, the old age convalescent plasma therapy is also proclaimed to increase the immunity to our fight against the virus. So here I want to bring about the the solution for the virus. So why I want to say this is because you have to be aware that you don't have to be a scientist or an accomplished scientist to bring the solution. So you all know about the smallpox, right? Smallpox was proclaimed as the death sentence, and it's a DNA virus in that case, and it was known to be originated long ago in 10,000 BC, and it was actually eradicated out of the world in 1979, the exact year when I was born. And so this was actually possible because one particular scientific discovery, not by that the idea was originated from a non-scientist dairymaid. So she observed along with others that I will not have smallpox because I've already had the cowpox. So she identified that when she is exposed to this pox virus, then the chances of her getting a smallpox is very less. Then the blood was taken from the dairy maid and was administered into the baby, and that's how a vaccine was born. So this was from the history of the germ theory of disease, the painting. So vacca means cow. So the term vaccine itself came from the etymology is from the cow. So because of this particular, just one discovery, you just imagine that the whooping cough, rabies, polio, chickenpox, diphtheria, tetanus are all kind of, now managed now. When I was young, I'm known to, the movie stars come in the television and say about the polio vaccine. And nowadays, you don't see polio is almost eradicated from India. And for SARS-CoV-2, there are a lot of vaccines that are under work. Either it can be an inactivated, virus, or this one particular synthetic peptide, laboratory-made piece of protein. So Moderna, I'll talk about that later. That has been showing to be very successful so far. Or it can be a recombinant viral vector, or it can be a DNA or RNA, carrying the pieces, of course, SARS-CoV-2. Again, neutralizing antibodies it's not exactly in vaccine. Antibodies like antibody technology, but it's also, in a way, kind of give that immunity. So it's also sometimes termed as vaccine. And also the virus-like particle, which has the same shape as SARS-CoV-2, and recombinant bacterial and vector vaccines, and live attenuated weakened viruses are also used. So these are all under works now, and a lot of development has been going on, like Picovac recently, or the Oxford vaccine. So they're all showing very promising result. Because my field is in nucleic acid, I just want to say only about the nucleic acid vaccine. This particular Moderna mRNA 1273. So what they do is a very elegant piece of work. What they do is, they don't make a complete, virus. So they just expose the piece of the, they just, overexpress the RNA, and then they try to, teach our body that, this particular virus is already known. So that particular, technology has been very, very useful, and it has been shown to be safe to the humans at this moment. And I'm counting on this particular vaccine, and it's expected to be released only in the endpoint, June 2021. So we have to wait again for the vaccine. So until then, we have to stay safe for our, survival. And this mRNA 1273 is the name, and so that is known to be special, very, very good for neutralizing the virus. And here I want to say the topic of this, particular webinar. So are these viruses are vacillating? Yes, they are. They are vacillating. That is very, very normal for any viruses, particularly an RNA virus. They tend to be misread because their mechanism of spreading is in such a way that all they need is just to infect a host cell and multiply themselves. They are mutating. That is true. There are a lot of strains, a lot of mutated strain that is known to be around the world. And but whether those are materially different. So when we say about mutation, we always tend to associate ourselves with this kind of X-Men or Fantastic Four or Thanos, right? It's not that bad. It's not the comic book kind of mutation which can lead to be a dramatically different thing. So far, whether there is a change in respect to transmissibility, virulence, antigenicity, or resistance. So that is the most key, right? Whether we need to make our vaccines to different viruses, different strains of viruses, or should we, should we not be worried? Should we be worried? Should we not be worried? As of now, the answer is we need not be worried as of now. Hopefully, we, I expect that, we need not to worry about, having several vaccines developed for this. Picovac actually say that, their vaccine is working for 8 different strains. But, as of now, there is not a proper consensus among the scientists that there are so many strains. At least they are not materially different. They're just, either silent or cosmetic. You have to know that the viruses is made up of 30,000 letters. And the mutation, just 10 letters or so is different among the viruses. There's a very small difference. Like, some people have a beard, some people don't have a beard, something just simple cosmetic changes are there. But whether they have a change in the virulence or antigenicity or transmissibility, not yet. So that is a very good news. That work was done recently published by Dr. Francois Ballox. So we have to be happy about that. So when the vaccine is going to be there, it's going to potentially is going to work for all the viruses. So here, the other thing I want to say is the lessons from nature. So this particular thing is that after 3.8 billion years of Earth, the survival, Earth is a standing proof. Earth always try to, irrespective of whether dinosaur is there, there's a meteor attack, life always finds its way. So they always cures itself. And this particular virus, I don't want to say this is good because a lot of people have died. It's a very, very sad thing. But this is one particular opportunity for us to, introspect ourselves, our way of living. Just like, the 2001 Space Odyssey, they talk about a monolith, that changes the, the perspective of a monkey, and then, the astronaut to go to a next level, right? So now we have to think about the next level. What you can see here is the deep park in Nara. This is kind of a sight straight out of heaven. So nature is finding its way again. So nature is reclaiming this space. So from now on, my point is we have to think about the sustainable coexistence. So we have to be very careful about our way of living to avoid this kind of viruses. So please try to live in a proper way from now. So cure will come, definitely. Cure means cure out, the etymology. The cure will come when we care about ourselves and our surrounding people and our nature. So please do that. So that's a take-home message I want to say here. And then I want to move on to my own work, which I will quickly say. So we are all made from letters, A, T, G, C. So as you know that, the A, T, G, C are shown in this kind of the DNA strands. So anything that is living, like either the bacteria, the the bug, which I was talking about, or the my baby. So all are made up of letters. Just that in the humans are made up of 3 billions of letters. So one thing is that these letters are stacked up in the DNA, which is 2 meters long. So how they get themselves, pack themselves into the cell, into the nucleus, which is just 6 micrometer. That's how, the living systems are controlled. So why do we look so different than a bacteria, than a banana? Banana shares 50% of our genome. So people have been thinking about whether the microscopic or the DNA makes the cell or the cell makes the DNA, but the actual control lies inside in the middle, the mesoscopic. So this particular mesoscopic is the around the nanometer range. So this is where the actual control exists. The way the DNA gets packed themselves inside the nucleus is how the gene is switched on and switched off. So I was talking about, this is the representation of how the DNA shown in the blue gets packaged themselves into the chromatin shown in the yellow, and then they get themselves get into the nucleus. And if it's going to be very difficult for you to imagine, then just imagine the letters. Imagine this DNA in form of letters. Just you can see in the ancient scrolls, right? Here you can see what is written here. Here you cannot see. So this is the on gene. This is the off gene. Let me explain in a much more simpler way. So just like a same paper, which is folded to form a different structure, to form the origami, the same way, the same genome can be folded in such a way that some of the genes related to stem cells can be switched on, and some of the gene which is related to heart cells can be switched on. So it can change case by case. So basically, we are all born out of the embryonic stem cells, so from out of the embryo. So which gene is switched on and switched off determine what kind of cells we have. So off gene is nothing but you can't see the natural copy machine, which is nothing but the RNA polymerase. They also cannot see, but when it can see, they can make a transcript out of that. So epigenetics is very important. A simple example I can say is that this is the picture of a queen bee and a worker bee. So they both started their life as the baby larva. So one consumed the royal jelly, and that opened up the chromatin structure. and then opened up the queen genes, so that became the healthy looking and bee. But the other worker jelly did not consume good food. So food is very important. So why I want to say here is we have to be resistant from now for coronavirus. So we have to have a very nice nutritious food. So you can see the effect of food just by the difference of the food. Worker jelly and royal jelly, the the longevity and the immunity level of the can be increased potentially. So this is the natural epigenetic code. I don't want to say more into chemical modification because this is more of a public lecture. So basically, this is the histone protein. A lot of modification can happen. So this can be methylation, acetylation, phosphorylation, and so on. And anyway, basically, the epigenetics is nothing but the layer outside the genome that determines whether the gene is switched on and off, in like I shown before, whether the gene can be read or unread. That's the point. So that can be, just imagine that as a script. Then there are certain enzymes like a writer, reader, and eraser. So that determine whether, whether the gene can be switched on and switched off. So that is determined by the enzymes like histone acetyltransferase, histone deacetylase, and so on. So there is a molecule called SAHA, so which is the HTAC inhibitor. Is a small molecule. This can actually open up the chromatin structure, basically stop the eraser, and then make the cell to write some of the genetic code to switch on certain gene. The problem with this particular molecule is that they can activate the target gene, but they can also end up activating the gene that is not supposed to be disturbed. So it can lead to severe side effect. So we wanted to know whether the PIP, which the molecule which I said that can read the DNA letters, to be the solution. So this is the video I'm showing. So we conjugated the sequence recognition part called pyrolimidazole polyamide, and then we conjugated that with the SAHA, the HTAG, the epigenetic eraser inhibiting part. So it goes and then it stops the eraser here, and that particular enzyme called histone deacetylase or HTAG. So when that is inhibited, the writer, the histone acetyltransferase, come and put a acetylation mark over the histone protein shown here. So in this way, you can actually remodel the chromatin structure or make the DNA accessible for the copy machine, the RNA polymerase, to come and make a copy out of this. So this is how under the complex environment, we were able to switch on the specific gene of interest. So this was kind of a very new technology which we have developed, and we were able to add the GPS to this particular keys, which is I'm just representing here. So this was, imagine this to be the genome of the human skin cell, where all the genes were silenced. Skin cell does not have the genes related with the retina or anti-cancer or so on. So, but we were able to activate the gene, the anti-cancer gene. We were able to activate the immune cell gene. We were able to activate the retinal cell gene. So you can see that all this molecule have the one thing in common, which is the functional part, that is the SAHA, which is the HTAC inhibitor, the epigenetic eraser or the writer. But just the sequence are different, by placing the pyrrole. This is the blue circle is pyrrole, and this is the red circle is imidazole. By just placing these two letters, P and I, in different places, we were able to send them to a different DNA sequence to switch on a specific gene of interest. So in this way, we were able to differentiate the stem cell into heart cell and into brain cell. And one work which we have done, that kind of that's my which I'm proud of, is that people, just imagine this particular lady, she's very happy holding a child. After a very long time, she conceived a child, she got a child, and she's very, very happy, and having a child in her hand, holding her hand. Just imagine the kind of trauma she will feel when she knows that that particular child has a rare disease. So this is, this is true that this can happen. This one of the syndrome is called Leigh syndrome. Leigh syndrome is caused by the defunct mitochondria, the complex one. So because of this, the mitochondria is the powerhouse of the cell, which I told you before. Probably more about that as the biotech students. So that can be present in all kinds of functional parts of our body, including the brain or heart and retina and so on. So wherever the mitochondria is present, whenever they are mutated, that can lead to this kind of very severe newborn. So in this case, it's very almost impossible to recover this kind of genetic disorder. So we kind of proposed a solution about that, and that particular proposal came not from me, but from my daughter. So my daughter is like me. She always asks a lot of questions, and so to stop her asking the questions, I gave her an a smart device, and then she used that smart device to learn some certain things. So one thing she said is that you are working on genetic switch, right? Yeah, because we call it as our technology as a genetic switch. I said yes. And she asked me, if that is a genetic switch, then can it work inside the powerhouse of the cell? Because then it's right. The mitochondria, as you know, is the powerhouse of the cell, and people always think the DNA, because we are working on a DNA-based genetic switch. People always think that DNA is inside the nucleus, but not many people know that the DNA is also present in the mitochondria. Even we, or I myself, I am not interested in mitochondria. Always I think about the DNA inside the nucleus. But this childlike brain, the children, they raise the curiosity, and then we thought about how we can make our molecule enter inside the mitochondria. This was kind of a mission impossible, because the mitochondrial membrane is kind of a fort with a lipid barrier and energy barrier. So we are not Tom Cruise, but we have a kind of student, Takuya Hidaka. He's kind of a very smart student, a brainy student. So he developed this technology, and we were able to have our molecule go inside the mitochondria and silence the disease-causing gene, ND6 gene, that is associated with this Lee syndrome. This was done only at a petri dish level. Now we are trying to advance this technology, but this was the first ever technology which can do this in the world. So one of the peer, he said this paper brought a big smile to my face because it's something I never thought of. But this technology was actually proposed by my daughter. So my point is, we have to have more scientists than engineers. Now we are, this is our startup company in collaboration with my boss. I owe most of my success to my boss, Professor Hiroshi Sugiyama, and we regulate genes as we want. And I want to end up in this seminar with this particular message. Again, we need more scientists than engineers. So please take research as your career, particularly after this coronavirus situation. The best of the brains, they tend to go into the engineering side, or to the HR side to make more money. But now, as you can see, that the money is irrelevant when we even the developed countries like Japan or USA, they are facing this crisis. So we need more scientists than engineers. So please take research as your career and try to ask why not. Why is very important. Why not can open up the new possibility, and to make you to see the unseen. We need to see the unseen. We need to know how to control this particular virus and the virus which may emerge in future. So thank you very much again for this opportunity, and thank you very much for your time. I'm ready for the questions now.
Yes, sir. Sir, thank you so much for the interesting and colorful information which you have given through your presentation, and also for sharing your research experience behind your inventions. And I am sure that participants would have got new insights through your session, sir. And it was very interesting to know about the history and about the recent types of vaccines and the works which have been going on right now. I've been getting questions being flowed up in our group. So I'll just go to the question session now. So participants, kindly post your questions in the live chat along with your name and your institution name. So the first question, sir, is from Sylvia from Bharathidasan University, Trichy. So our question is, what are the factors that make the COVID cases vary between the male and female population?
Yeah, that is a good question, and people are still finding out the answer. So one of the still is a new virus, so we can only hypothesize at this moment. So one of the hypotheses is that the chromosome, the difference between the the male and female chromosome, that is one of the reason. The other cases they say is that this cannot be entirely true, but relatively the smoking, people, the men smoke a lot than the women, so the chances of their lungs getting this kind of susceptible towards the COVID is also high. So at this moment, the more reasonable thing could be because of the chromosome, and so that is the current hypothesis. It's not I don't think there is not enough literature, solid literature, which can validate this hypothesis. But this is the current hypothesis.
Okay, thank you, sir. The next question is from Annie Aparna from WCC College, Zoology Department. Her question is there any other faster way of detecting the presence of virus other than X-rays and PCR?
There are a lot of, see, the point is detection. Detection is the most important key step to treat this, right? So the main problem of this particular COVID-19 than the previous counterparts like SARS and MERS is that the symptom is not clear. Some people remain asymptomatic. What is the symptom? So whether the fever is a symptom or cough is a symptom. So many people, depending on the viral load, depending on their immunity, people are not showing the symptoms properly. So it is very, very difficult to say, have a proper technology which can determine. For example, I know that people have been using this AI, even the cough, the voice, the way people speak, or the way people cough, or the other kind of small changes can also be shown, interpreted as a symptom of coronavirus. So in that aspect, yeah, there are certain technologies which are under development, particularly the LAMP PCR. Even the PCR, there are certain new kinds of PCR which can give the answers much faster, irrespective of the strain variation. So a lot of things are there, but at this moment, PCR is the most reliable technique. Other than that, even the the rapid antibody test kit, probably that it's not so reliable.
Yeah. The next question is from Shalini Das, Jebas College. Our query is how SARS-CoV-2 different from MERS genetically and other aspects also?
Yeah, there are very simple. The letters are different. Basically, as I told, at the fundamental level, there is a difference between the genome. It's not entirely different, a very small difference, and that particular difference, the materially it became different. Materially, it became different in the sense there are two major things which made this to be more infectious towards human. One is the way they attach to the ACE2 receptor. The host cell, they have the receptor, the ACE2 receptor. So the attachment is much better, much stronger, and the binding affinity is much stronger than the SARS-CoV or the MERS-CoV. And then the budding process, the way the virus buds and then releases genome, that is also different. So these two are the major differences than their early counterparts.
Okay. Next question, sir. It's from Pooja Shri, Adiyaman College of Engineering. She's asking a question: can a same person be affected by coronavirus twice? About the reinfection of coronavirus.
Yeah, one good thing is not now. So there has been reports which were saying that some reports which were saying that in Korea the people have been detecting the reinfected situation, but no, that was the dead virus particle that they detected in that test. And as of now, people who are getting infected by the coronavirus are not getting infected again, but this is still too early. We have to wait until next 1 year or so to know whether there is any chance of reinfection that can happen or not. The reinfection can be because of two things. One is by the different strains, which so far is not possible because there are not many different strains as of now. So the other thing can be the virus can be in our system, hide in our system and then get re-exposed when the time comes. both these things are not happening as of now. So as of now, no, it cannot. So people who are coming out of virus at this moment can be considered as the corona-free and having immunity against that.
So next question is by Saurav Kumar from MITS School of Biotechnology. Is any gene responsible for COVID-19 being non-symptomatic?
What is non-symptomatic, right?
Any gene responsible for being non-symptomatic?
We can't say, see, it's very difficult to say, non-symptomatic. So gene in virus or in human. So, okay, I'll tell you, because probably this is a live chat. I don't want to re-ask the question. I'll answer for the both. So see, in human, these asymptomatic is because of two major things. One is the number of viral load, how much virus one intake during the course of infection. The other is the level of immunity. When you say immunity, it doesn't mean that the immunity is strong or something like that. In some cases, the immunity is very much controlled. And also the virus, in terms of viruses, whether the virus, the budding and the releasing of viral particle and the copying of the viral particle, whether all of them are in a proper way or not. Like I said, this is a vacillating, like in this particular topic, the vacillating virus. This can under the whenever it gets copied and then they try to incorporate themselves in the host genome, they can have some variation. That can also be one of the reason. But the asymptomatic is still not clear. People still don't know what is asymptomatic because still we don't know the clear symptoms of coronavirus. Whether fever and cough is the only symptom, major symptom, but is it the only symptom? We don't know. It's too early to say. Yeah, but there are some genes which is responsible.
Okay. Next question is from Nismambi, Bharatiyar University. The question is, in vitro mutagenesis to create positive changes in COVID-19 gene, how far this can be possible?
Yeah, see, at this moment, the most better thing at this moment can be towards the vaccine. Of course, all these kind of very nice technologies are there, like in vitro mutagenesis or antibodies and so on. But how much it is possible for us to incorporate, actually do for the human, we have to be very careful. Why people have been very careful in not developing a medicine and announcing a medicine is because we don't want to end up messing our system, right? So we have to be very careful in not messing up our system with the the technologies which is still under there's a very nice technology, very fancy technology. It's a very good technology, of course, I understand, but whether that can have some side effect is not known. So we have to be very careful in those kind of technologies.
So next question is by Ms. Sandra Vistas. She's asking whether asymptomatic carriers of COVID are more immune towards the infection.
Yeah, see, again, it's a very tricky question, and to be honest, I cannot say very clearly, but what I can say here is that they are not more immune. Their immune response could be, different from others, but still some of the asymptomatic carriers are known to be super spreaders. So it's not actually affecting our organs as of now, the carrying capacity, but it's also spreading. So it's very difficult to say. Immunity means, yeah, the response is less, but whether they are more immune, I don't know.
Next question is by Taslim. There is a myth that COVID-19 is a chimera of pangolin and bat. Is that true?
It is a chimera of, at this moment, it is, as of the paper which was published in Nature, it seems to be the chimera, the recombined virus of the pangolin and the bat. But I think the question is about whether it's man-made or not. Yes. So we have to be very careful in this case that whether it's to say anything is man-made or not. As of now, there is no proper proof which says that this is man-made. As of now, this indicates only the natural selection. Why the question of man-made came is because this particular virus has already reached the peak. Whenever the mutation happens, the mutation is either can be positive or negative in terms of virus. The virus wants to multiply themselves, so they want to be the best in the best scenario to attach the host and multiply themselves. And this particular virus is in that particular primed state. So that's why people have been wondering whether this can be naturally occurring, or maybe this is man-made. It's too good to be naturally selected. But as of now, the research indicates only the natural selection. So please don't go by any myth which is stating otherwise. Unless and until we have a solid proof, we are not supposed to say the conspiracy theories at this moment.
Our next question is by Kalai Kavi. Can the organic medicines will cure and prevent from the COVID-19? Is it effective?
Okay, so organic medicine in the sense, I suppose it's about the Siddha medicine or
yeah.
so on. Yeah, traditional medicine, right? So in China, they use the traditional Chinese medicine. So even when we have the fever or cough or whatever, so there's not only one medicine we use. We also use medicine, but we also eat the rasam rice or whatever the natural thing to make ourselves feel better, right? So basically all this kind of traditional medicine or food is basically medicine actually in this case. So all this kind of traditional Indian medicine or traditional Chinese medicine, they are all good for as a side as a kind of not exactly placebo, but it makes you feel more comfortable. It makes you to avoid the fever, try to in a way control the immune response. So it is additive effect, but it's not a standalone. As of now, there is no traditional Indian medicine or Chinese medicine or whatever the medicine known to have a standalone effect on coronavirus. So this can be useful. You can take it if you feel comfortable, but you have to do under the prescription of the prescribed medicinal, like Siddha medicine people or something like that. Ayush at this moment, I think they already announced the Kabasura Kudineer. You can take that, but please take it in caution. And to answer this, as of now, there is not enough evidence which can say that this is a standalone, but it can be an additive effect. Yes.
Next question is by Yashashwini from WCC. Is plasma therapy effective in treating COVID-19? And this effectiveness vary from one population to other population, the success rate?
Yeah, plasma therapy is an age-old technique. Of course, it is looking to be very successful at this moment, but we have to be very careful. Again, all this technology, there are two things which I came to realize when I do this X-ray technology. Doing a good science, a great science, or doing something which is useful. Okay, so these are all the very, very nice, exciting technologies, like but when it comes to applicability, how can you think about having so much of plasma out of the patient, right? So much of plasma, enough plasma to treat the patient. It can be successful. It is successful, of course. It is doing a wonderful job, and the monoclonal neutralizing antibody is also very, very good. But can we scale it up to available to all the population? So that is under question mark. And there can also be a potential side effect, right? So we have to be very careful in that aspect.
The next question is from Janet Niveda, WCC. So can conserved sequences in viruses be used as a target for all the mutated types of COVID-19 virus?
Yes, it is. Yes, it is. It's a very good question. And can I have the name again?
Janet from
Janet.
WCC. She's from our department, sir.
Oh, good question, Janet. That's a very good question. And that's what we are working on, actually. So it's good that you thought the same. And I think there are several conserved sequences which we can target. And, usually the one beautiful thing about any nature is that they use minimal material. I mean, to make, just like I said, just the same DNA, it made so much of living things, right? So, that kind of conserved sequence exists. So many are there, and we ourselves are also thinking about targeting them. And yeah, that is possible too.
Next question is from Pavitra, WCC. Is this immune boosting foods are sufficient in this virus attack, or we need to take any supplements for its prevention?
Yeah, so again, immune boosters and supplements again. See, it became these days, because of the WhatsApp University, A lot of things are going around, and it's very, very difficult to say. Immune booster is a abuse term again. We don't need to increase our immunity. Most of our drugs which have been used are basically immunosuppressive drugs. We need to control our immunity. And in that aspect, of course, this immune controlling food, prophylactic, supposed to be prophylactic, like kabasar kudineer, or the vitamin C or whatever, that is supposed to be good enough, good enough in a way that providing nutrients and enough nutrients for the body to attack. But yeah, it is not sufficient. We need to have controlled effect. We have to be happy, right? We are basically, man is a social animal. So not just the food, our environment, our happiness, everything is interrelated towards the immunity. Yeah, so we have to be happy and healthy and confident about ourselves.
Okay. So one last question, sir. With this, we are closing the question answer session. So when will this pandemic come to an end? Will it end, or we have to adapt to live with this virus?
Yeah, so that is a question I'm asking myself. So yeah, so my perspective is that it's difficult to adapt. As in Sweden, the herd immunity is not working properly, so it is very, very difficult. So this pandemic will come to an end, that I'm very sure. I think vaccine will be found, is my perspective. I'm not talking out of my thing as a scientist. It's my own perspective. This pandemic will definitely come to an end in a year or two, I guess. But we will be in for more viruses. So we have to be very careful. This is natural selection. It is more dangerous because it means that more this kind of viruses can happen. So we have to mend our ways. We have to start thinking as a, the Neil deGrasse Tyson says, we have to start thinking as a species. We have to be mindful about us, to others, and to the environment. And in this, we have to be prepared that, for this virus and the future virus. Adapt means we have to adapt for preventing, and hopefully for the future also we need to be mend our ways.
So it's the question that is lingering in most of our minds, sir, at seeing the present situation. So thank you,
sir. Overcome this. Yeah,
sure. So thank you, sir, for patiently addressing the questions of the participants and putting very clear explanation in the way which they can understand. So now we have come to the close of the session 1 of webinar series. On behalf of Department of Biotechnology, Women's Christian College, Chennai, I wish to extend my heartfelt thanks to our resource person, Dr. Namashivayam Ganesh Pandian, for his presentation, which was loaded with interesting information. We are grateful to you, sir, for taking your time off to share your experience with us and patiently answering our questions. We thank all our participants for staying with us throughout the program and bombarding us with questions. So it is indeed an encouraging gesture for us. We extend our sincere thanks to our principal, Dr. Lillian Jasper, and the WCC management for the constant support. We wish to thank our student volunteers who have stood by us throughout the conduct of this seminar. Thank you all. And we request you to join us again on Wednesday, 27th May 2020 between 10:30 a.m. to 12 noon for the next webinar series given by Dr. Ashok Kumar P, Associate Professor, Department of Genetic Engineering, Madurai Kamaraj University, on diagnostic tests for COVID-19 daunting challenges. So stay tuned in our YouTube channel for the updates. Stay safe. Wear mask, and let's fight COVID-19 together. Thank you. Thank you all once again. Have a nice day.